All right, let's now talk about gene linkage. Gene linkage is the term we use to describe two genes which are found on the same chromosome. Therefore, they tend to be inherited together, and the rules of independent assortment don't necessarily apply. Let's consider a diploid cell, which has two sets of homologous chromosomes. So in this instance, I'm going to draw my two sets of chromosomes. They contain on them the gene G and the gene N. Now, during meiosis, the homologous pairs segregate from one another, effectively separating the two copies of the gene, the two alleles, and also the sister chromatids separate from one another. This is due to that principle of segregation, so that in my end results, I end up with 4 haploid gametes. If I was to consider this, I would end up with 2 gametes which have the capital G, capital N chromosomes times 2, right? Because the sister chromatids segregate as well as the homologous chromosomes. And I would have these 2 chromosomes over here with the lowercase g, lowercase n, and I would have 2 cells that look like this. So the genotypes of my haploid gametes would be capital G, capital N, and lowercase g, lowercase n. Is this my only possibility? No, it's not my only possibility, because we also know that due to independent assortment, these chromosomes may line up in another way. My lowercase n may line up on the same side as the capital G, and my capital N may line up on the same side as my lowercase g, so that when the homologous chromosomes are pulled towards the poles, now I have the capital G gene going with the lowercase n, and the lowercase g gene going with the capital N. The end result would be 4 haploid gametes, the capital G and lowercase n being sorted into the same cell, and lowercase g, capital N being sorted into the same cell. Again, each of these times 2, because the sister chromatids also segregate. And I would end up with the following genotypes: capital G, lowercase n, lowercase g, capital N. Any of these have equal probability of appearing. There's equal probability of each of these gametes being formed. In other words, it's a 1 to 1 to 1 to 1 ratio. So if I were to look at the offspring from this parent, I would notice pretty much equal proportions of these 4 different gametes and the 4 different phenotypes showing up because of this. If I were to cross this with, say, a homozygous recessive parent. The example I've given you right here is where the genes are not linked. So there's no gene linkage in this example. The genes are found on totally separate chromosomes.